Hello and welcome to Microbiology Mentor. Today we will be discussing one of the most important bacterial families, the Enterobacteriaceae, and its general characteristics feature. This family is also of historic importance because it includes plague bacilli and typhoid bacilli, the most important bacterial pathogens in human history. So this family is one of the largest, most heterogeneous collection of medically important gram-negative rods. It includes almost 50 genera, hundred of species and subspecies, and this classification is based on various factors like biochemical properties, antigenic structures, DNA hybridization, and 16S ribosomal RNA sequencing. Despite of its complexity, most human pathogens are caused by relatively only few species. Let's see the presence of these microorganisms. The member of this family are known to be ubiquitous worldwide in soil, water and vegetations, and also they are known to be the normal intestinal flora of most of the animal as well as humans. The member of this family also causes a variety of human diseases in various ways, while some of the members are almost always associated with human infections like Salmonella, serotype typhi, Shigella species, and Yersinia pestis. Other members like E. coli, Klebsiella pneumoniae, Proteus mirabilis are the members of normal commensal flora, but that can cause opportunistic infections under certain situations. So let's see the definition of Enterobacteriaceae. The definition of Enterobacteriaceae are on the basis of certain characteristics, almost 10 of them. So let's see one by one. So the members are gram-negative rod, do not produce any spores, motile by the presence of peritypous flagella. However, there are certain non-motile members like Klebsiella, Shigella and Yersinia. They are known to grow on ordinary laboratory media, grow both aerobically and anaerobically without any additional supplements. They are known to be active biochemically and ferments D-glucose and other sugar. They are known to be catalase positive and oxidase negative. So the absence of cytochrome oxidase activity is an important characteristic because it can be measured rapidly with a simple oxidase test, which is used to differentiate the Enterobacteriaceae from other fermentative and non-fermentative gram-negative rods like Vibrio and Pseudomonas. The members also reduces nitrate to nitrite and contain enterobacterial common antigen ECA. At the same time, the GC content of the members of the DNA is typically 39 to 69%. Now let's see some of the common medically important members of the Enterobacteriaceae. Here I have listed some of the common members alphabetically for the sake of convenience. Citrobacter fundei, Citrobacter kozeri, Enterobacter aerogenes, Enterobacter cloacae. The commonest is Escherichia coli, Klebsiella pneumoniae, Klebsiella oxytoca, Morganella morgani, Proteus mirabilis, Salmonella enterica, Serratia marcescens, Shigella soni, Shigella vaccinari, Yersinia pestis, Yersinia pseudotuberculosis and Yersinia enterocolitica. Now let's see the antigenic structure in general of the Enterobacteriaceae. The antigenic structure is a complex one and consists of somatic O, capsular K, and flagellar H antigen as it is shown here in this schematic representation. And if you just see the enlarged form of this presentation, here you can see this is a flagellar antigen, capsular K, and somatic O antigen is the outer part of the lipopolysaccharide. So the members can have more than 150 type of heat stable somatic O antigens, more than 100 of heat labile K, that is capsular antigen, and more than 50 H or flagellar antigen. Here the audience must make a note that in case of Salmonella typhi, the capsular antigens are called VI antigens rather K antigens. Now let's see the various characteristics of the antigenic structure like O antigen first. So as we have seen that O antigen is the external most part of the cell wall of the lipopolysaccharide. They have a characteristic sugar, resistance to heat and alcohol, and are usually detected by bacterial agglutination. Each genus of Enterobacteriaceae is associated with a specific O group, although a single organism may carry several O antigen. Most Shigella share one or more O antigen with E. coli, and that is the reason for the cross reactivity between the Shigella and E. coli. Also, E. coli may cross react with some Providentia, Klebsiella and Salmonella species. O antigen may be associated with specific human diseases, and specific O type of E. coli are found in diarrhea and urinary tract infections. Now coming to the K antigen. External to O antigen, on some but not all members, K antigens are present. In nature, they are basically proteinaceous, but some may be polysaccharides, including the K antigens of E. coli. This K antigen, when present outside, will interfere with the agglutination by the O antisera. This K antigen may be associated with virulence, like E. coli strain producing K1 type of antigens are predominantly seen in neonatal meningitis. At the same time, K antigen of E. coli causes attachment of bacteria to epithelial cell prior to gastrointestinal or urinary tract invasions. Now continuing K antigen, Klebsiella form large capsule consisting of polysaccharide K antigen, which can be identified by capsular swelling test with specific antisera. Specific type of K antigen may be associated with specific conditions like respiratory tract infections are caused by capsular type 1 and 2, and urinary tract infections by type 8, 9 and 10 and 24 of the K antigen. Now coming to the H antigen, as we have seen that H antigens are located on the flagella and are denatured either by heat or alcohol. However, this flagellar antigen can be preserved by treating with formalin. Within a single serotype may be present either in one or two form, which is called phase. So there are phase variations. The organism tends to change from one phase to other, which is called as phase variation. This phase variation may be important as they may invade the immune response developed by the host cells. Now let's see the common virulence factor associated with the Enterobacteriaceae. The first one includes the presence of endotoxins. So the activity of this toxin depends on lipid A component of the lipopolysaccharides. Remember that the lipopolysaccharides, the inner part is the lipid A component, which is released when the cell get lysed. Many of the systemic manifestation of the gram-negative bacteria are mediated by this endotoxins. Activation of the complement, release of cytokines, leukocytosis, thrombocytosis, disseminated intravascular coagulations, fever, decreased peripheral circulation, and death may occur because of the release of the endotoxins. Other important virulence factor includes the capsule. The presence of capsule is a virulence factor because it prevents the phagocytosis. The encapsulated Enterobacteriaceae members are protected by phagocytosis by the hydrophilic capsular antigen which repel the hydrophobic phagocytotic cell surface. Now, other common virulence factor includes the antigenic phase variation, which I discussed before this slide, where the flagella can be present in different phases, and this phase variations can be seen and they may invade the immune response because of the presence of the different phases. Now, other common virulence factors of the members of Enterobacteriaceae includes the presence of type 3 secretory system. What is this type 3 secretory system? Type 3 secretory system is a molecular syringe. It is consisting of approximately 20 proteins that facilitates the transfer of these bacterial virulence factor. All those virulence factor that are produced in the bacteria, they helps in the transfer of these factor into the targeted host cells. Now, the other virulence factor can be sequestration of the growth factor. So, we all know that the bacteria needs certain growth factor in form of iron, which can be important growth factor for the growth. But this iron is bound in the host in the form of heme protein like hemoglobin or myoglobin, or it is bound to some chelating agents like transferrin or lactoferrin. So, what does this microorganism does? Because of the presence of some virulence factor, the bacteria counteracts the binding by producing their own competitive siderophores or iron chelating compounds. Now, this iron is now free, available from the host by hemolysis, and the microorganism can easily use it. Now, other virulence trait can be resistance to serum killing. As we know that in serum, the microorganism when present in the blood get easily cleared by the blood because it is capable of producing, a microorganism is capable of producing a systemic response. So, the blood tries to evade these, but the members of Enterobacteriaceae have develop resistance to this serum killing. So, this is acting as an important virulent factor. Now, antimicrobial resistance, various antimicrobial resistance determinants as well as antimicrobial resistance against different drug and drug class also act as a common virulent factor for the microorganism. Now, with this, I come to the end of the general characteristic feature of Enterobacteriaceae. Thank you.